Fresh frozen tissue is the gold standard for studying the brain, especially when you're dealing with fragile RNA transcripts, signaling proteins, or rare human samples. By enabling high-plex RNA and protein detection on the same fresh frozen section, COMET brings together information that previously required separate assays, separate workflows, or simply wasn't possible. The majority of the spatial biology platforms today only offer spatial transcriptomics or spatial proteomics, but cannot detect both on the same tissue in a high-plex format. Moreover, other technologies may allow limited multiplexing, but require multiple rounds of staining, lengthy workflows, image stitching, or complex manual steps. COMET delivers true high-plex spatial multiomics in a single automated workflow, dozens of RNA and protein targets, same slide in one experiment. Together, they define precise self-states which are involved in neuroinflammation, neurodegeneration, remyelination. RNA tells you what the cells plan to do. Protein tells you what the cell is actually doing. Together, they provide a systems-level understanding necessary for deciphering neural circuits and disease networks. Using spatial multiomics on COMET, researchers can now correlate transcriptional states with protein signaling, receptor activation, or cell-cell interaction within the tissue context. You get molecular phenotype and functional phenotype simultaneously, which is transformative for understanding neurodegenerative disease pathways. In Alzheimer's disease, for example, microglia have dozens of activation and disease-associated states. Many states cannot be defined by RNA or protein alone. COMET enables simultaneous visualization of transcriptomic signatures, for example, looking at the TREM2 gene, SPP1 gene, as well as functional protein markers for microglia, for example, EBA1, CD68. Another great example would be looking into brain tumors and glioblastoma research, which can reveal tumor-immune interactions, heterogeneity in signaling pathways, and looking at oncogenic transcripts and protein markers simultaneously. Traumatic brain injury is another example where you can assess the transcriptional response to injury while measuring cytokines, neuronal viability markers, and repair proteins all within the same tissue. Neurodevelopment and psychiatric disorder is another use case where fresh frozen compatibility enables work with delicate developmental tissues, allowing the study of RNA transcripts and protein-level circuit markers in the same experiment. If you want a platform that lets you analyze brain complexity at the level it deserves, transcript and protein, cell and structure, discovery and translational workflows, COMET brings them all together. Spatial multiomics isn't the future anymore, it's happening now, and COMET is the platform built to deliver it at scale.